Good evening, everyone. My name is Bruce Christensen, and I am the director of the Research School of Psychology at the ANU, and it's my absolute pleasure to welcome you to Psychology's 2022 annual lecture by the University of Minnesota's Distinguished McKnight University Professor Bob Krueger. We are meeting today on Ngunnawal land, the sovereignty of which has never been ceded, and I would like to pay my respects to Ngunnawal elders past, present, and emerging, and acknowledge their sage and committed stewardship of this place. I would also like to celebrate the positive influence of their culture here and across wider Australia for past millennia and ongoing into the future. There are a number of staff who have worked very hard to organize this event, and I'd like to thank them. Our head of alumni and engagement, Dr. Erin Newman, has been the chief organizer of this event, and the RSP is very grateful for her hard work and expertise in bringing this together. Our administration team, including Matthew Boyd, Karina Bird, Cheryl Morse, and Jonathan Meyer-Walkers, were indispensable in bringing the details together. And the college's marketing and communications coordinator, Barb Karapi, out here somewhere, great, very ably got the word out and made everything look nice. So thanks everyone for their efforts. And I'd also like to thank and congratulate the students who presented posters at our reception. Today's lecture is another in what has emerged as a strong and important tradition for psychology at the ANU. It started in 2015, and this is our 7th lecture. The purpose of these lectures is to invite outstanding and world-leading scientists, practitioners, and/or decision makers to deliver a lecture to both our university and wider ACT community that highlights an increased understanding of and meaningful impact on the world around us through psychology. In 2015, former Australian of the Year and Origin and Headspace founder Pat McGorry delivered a lecture on early intervention and youth mental health. In 2016, Tim Soutphommasane, Australia's race discrimination commissioner, talked about the challenge of social cohesion, especially as it applies to multiculturalism. In 2018, professor and the first Aboriginal psychologist in Australia, Pat Dudgeon, delivered her lecture regarding different paradigms for Indigenous mental health in Australia. In 2019, Professor Elizabeth Loftus, noted in the Review of General Psychology as the 58th most eminent psychologist of the 20th century, gave her lecture on the fiction of memory. And in 2020, the University of St. Andrews professor and social psychologist Stephen Riker spoke about COVID-19 and the contribution of psychology to understanding people's behavior under crisis. And finally, last year, we had Professor Jane Halton, who's recently been appointed as an honorary professor in our school, and who's held several prominent positions in government, including Secretary of the Department of Health and Aging, gave her lecture entitled Prevention, Preparedness, Politics, and Pandemics, Where Does Psychology Fit? Tonight, this tradition continues, and we're delighted to welcome another esteemed psychologist, Professor Bob Krueger, to talk about the important impact of psychology in contemporary society. And arguably, we're living at a time that needs psychology more than ever. It's a time with new and formidable risks and consequences. Climate change, rising mental ill health, violent extremism and war, growing wealth inequality, and global pandemics, just to name a few. Each of these has at its core psychological consequences and psychological explanations. And the solutions to each of these call for psychological action. Now, to be sure, technological, political, financial, and biological solutions are also guaranteed to play a role in tempering the tide of these grand challenges. But psychology will also be key. A key to positive behavior change and pro-social action. A key to developing communities where members belong and feel valued. A key to making hard, forward-thinking decisions. A key to fostering relationships built on trust, respect, and compassion, and a key to building resilient and flexible societies that can adapt in the face of unanticipated change. There are also reasons why psychology is well positioned, presently and in the past, to help our communities meet the challenges of the 21st century. But chief among these is psychology's long and proud tradition of expressly combining strong science, strong compassion, and strong action. And today's lecture will explore one of these knowledge traditions. And this tradition is poised more than ever to foster great insights into human experience, function, and health. This area is behavioral genetics, how genetics and the environment combine to give rise to individual differences. And there really have been few areas of science more fiercely contested. However, the idea has been around for a long time. Plato talked about the heritability of personality characteristics, and it was described by Shakespeare as a dual influence, that of nature and nurture, in The Tempest, where he wrote a devil, a born devil, on whose nurture can never stick. And at the end of the 19th century, polymath and British scientist Francis Galton, deeply influenced by his cousin Charles Darwin, worked tirelessly to empirically quantify heritability across psychological processes. Now, in the early part of the 20th century, the blueprints for understanding human experience, behavior, and cognition shifted from biological explanations. The rise of behaviorism and psychoanalytic theory pointed to the environment and intrapsychic conflicts and processes, respectively, as the forces shaping human psychology. Although a steady trickle of scientific inquiry focused on behavioral genetics throughout the first part of the 20th century, it wasn't until the 1960s and the advent of research methods designed to remove the confounding of genes and environments, designs such as pedigree and family studies, twin studies, adoption studies, that behavioral genetics cultivated renewed scientific interest. These advances launched a fertile and contentious period in psychology where genetic versus environmental causes of individual differences were debated, fought over, and elucidated. More recently, substantial innovation in the molecular analysis of genotypes and the biological processes governing gene expression, so-called epigenetics, have offered a new toolbox to fuel additional findings. Now, collectively, these lines of research have produced several powerful and exceptionally replicable conclusions about the impact of genes on behavior, which was recently summarized in a Perspectives on Psychological Science article by famed behavioral geneticist Robert Plomin and colleagues. For example, we know now that all psychological traits are under substantial genetic influence, but we also know that no human traits are 100% heritable, and that, in fact, environmental influences tend to make members of the same family more different rather than more similar. We also know that heritability is caused by many genes and that the stability of psychological traits across the lifespan is owed to genetics. Interestingly, the heritability of many traits, such as intelligence, increases as we mature. That is, the impact of genes on intelligence is greater in adulthood than it is in childhood. And finally, we know that psychological disorders are not qualitatively different from the normal range of behavior and experience, but merely result from the quantitative extremes of the same genetic environmental factors that affect everyone. This has led to significant shifts in the ways psychological disorders are conceptualized and to the realization that characteristics of psychological problems are common across the human population and differ in amount rather than in kind. Importantly, though, this research has also identified the prodigious impact of one's environment and experience in shaping human psychology. For instance, the importance of social relationships and group processes, childhood attachment to caregivers, behavioral modeling and reinforcement, practice and mental stimulation, self-reflection and metacognition, and so many more. In fact, the field has now shifted from its obsession with demonstrating the superiority of either nature or nurture to understanding the importance of both and to gaining a sharp insight into the interactive and synergistic nature of these forces. Today's lecture is very squarely in this domain. Now, it's important to also realize that there are very real possibilities that prioritizing genetic explanations may unnecessarily lead to a deficit model, where problems are often considered to be immutable, enduring, and stagnant. Among the more odious expressions of this perspective have been in the past systematic oppression and maltreatment of people with specific ancestry or disability on the grounds of genetic inferiority. Psychological research has a role in these ideas, and the field is making a renewed effort and promise to ward against the negative impact and misuse of such findings and reinforce the value of all and the dignity of all. At the same time, it's important that psychology continues to grapple with challenging results of scientific inquiry, yet in a sensitive and respectful manner. I believe that the work at the intersection of social and biological determinants helps put the field in better stead and underscores the adaptive and dynamic nature of human behavior and experience and its continuity across differences that create a very rich tapestry. So we are very fortunate to have Bob here to discuss this intersection. He completed his undergraduate and graduate work at the University of Wisconsin and his clinical internship at Brown University. His major interests lie in the intersection between psychopathology, personality, genetics, health, and aging. Early in his career, in addition to his genetic interests, Bob came to realize that the classification of mental disorders was key to understanding the genetic and social causes of the development. This led to quantitative modeling, and quantitative models proposed as alternative frameworks for understanding, measuring, and conceptualizing psychological disorders. In particular, this work has fundamentally moved the field away from classification by discrete categories, disorders, for example, to viewing psychological problems as a manifestation across a series of important dimensions that are contiguous with a wide array of normal experience. This work has been notable for its empirical and computational rigor and for pushing the fields towards evidence-based assessment that extends beyond observation and opinion. It has allowed, as we will see tonight, for refining our understanding of the biological and social causes of psychological problems and health more generally. Bob's work is also developmental, and he's leaned on the power of longitudinal designs and analyses to uncover the interplay of these effects over time, including among individuals who share varying amounts of genetic and or environmental similarity versus distinctiveness, twins versus siblings, for example. Bob has received several major awards, including the University of Minnesota McKnight Land Grant Professorship, the American Psychological Association's Award for Early Career Contributions, the Award for Early Career Contributions from the International Society for the Study of Individual Differences, an American Psychological Foundation Theodore Millon Mid-Career Award, and the Hoke Award from the American Psychological Association, or American Psychopathological Association, excuse me. He's been named the Clarivate Analytics, highly cited research for several years running, and research.com ranks him in the top 100 most impactful psychologists in the world. He's also the editor of the Journal of Personality Disorder and served as the chair of the US NIH CSR Social and Personality Interpersonal Processes Grant Review section. It is my pleasure to welcome him here tonight to deliver his talk entitled Nature, Nurture, or Both: How Biology and Experience Impact Mental Health.
Thanks so much, Bruce, for that very kind introduction, and it's a great pleasure to see so many people here today. I realize it's Friday afternoon, and I've prepared a somewhat academic lecture, so we'll see how that all goes. But hopefully I can give you some, exposure to the kind of work that we do in this, zone that Bruce was referring to. And I think it's accurate to say that the thing that I've been interested in throughout my career is how to integrate nature and nurture, how they're both important, and how that's sort of, in some ways, obvious, right? It's pretty clear as you look around you that it can't be the case that individual differences in human behavior are solely genetic. That wouldn't make much conceptual sense, because what do organisms do, right? Especially human organisms, they perceive and interact with the environment, and oftentimes for humans who are very social animals, the social environment. So I think there's lots of reasons to think in sort of an a priori sense that both of these things must matter. But the challenge, of course, is there a scientific way to understand this interplay? So it's one thing to say, it seems like both are important. It's quite another to say, these specific genomic polymorphisms are important, or these specific psychological processes emerge under these sets of circumstances. So a lot of our research is designed to kind of unpack that connection between genetics and environmental influences. So today I'm hoping to go through a little bit of concepts, strategies, and designs in behavior genetic inquiry. So the kind of field that, that Bruce was referring to, that a lot of the work that we do can be thought of as being located in. I'll talk about an example of the importance of G by E interaction. So the idea that genetic influences are not invariant across different environmental exposures, right? So that's a kind of a key paradigm for understanding where the intersection might exist, so that there aren't these sort of indelible, inflexible kinds of genomic influences, but rather that their impact varies substantially as a function of the environment in which people find themselves. So I'll talk about some of that work, and then I'll talk a little bit about sort of frontiers in this area. So the recent molecular frontiers in this area are nothing short of astonishing, even in the course of my career. So I've had a PhD since the late 90s and have been working, in this field. I could not have predicted the changes in technology that I've witnessed. The Human Genome Project was a remarkable achievement. It leads to the ability to take, modest samples of biological tissue from humans and basically survey and query the entire genome, right, from front to end in a human being. And that's an astonishing sort of technology in a lot of ways, almost sort of science fiction-y in some ways, at least in terms of my reaction to that technology when it became available, and also yields important conceptual and ethical questions that we're going to need to struggle with. But as Bruce sort of alluded to, if we don't struggle with those questions, the technology will get away from us, right? And so I think it's critically important that we both embrace how remarkable what we can now do in a scientific sense is, but also try to pay close attention to how to use that technology responsibly and in the service ultimately of human health and well-being. Okay, so a little bit about kind of twins. So this is a sort of textbook kind of description, but for those of you who aren't familiar with the biology of twinning, it kind of underlies a lot of things that we're able to do. Twins are a remarkable experiment of nature. They come in two basic varieties. There are identical twins and fraternal twins. So identical twins emerge when a single egg is fertilized by a single sperm and then it splits in two. So in this sort of cartoon representation here, you have a womb that's fertilized by a single sperm. And so you end up with two human beings that have the same underlying nucleotide sequence. So I'll say more about what that means. Actually, if you wander over that direction, you'll see the 4 nucleotides on the wall, which is kind of interesting because there's a sequencing center just on the other side of this room. So there's a lot of very high-tech genomic technology right over there. So that's kind of interesting as a juxtaposition to this talk. And fraternal twins, right, by contrast, those are separate eggs that are fertilized by separate sperm. Fraternal twins share only about half their genes, independently segregating on average. I have to add that language for, the hardcore geneticists in the room. But roughly speaking, you can think of it as 50% of their genes that vary from person to person are in common with each other. That's the same sort of coefficient of genetic relatedness that you see in siblings. The difference in fraternal twins is they're like siblings that were born at the same time, essentially, or conceived at the same time, roughly speaking. So that's an interesting set of, comparison persons to identical twins, because these different kinds of twins emerge from these very distinctive biological processes that result in human beings that are, distinct in their genetic resemblance. Here's another illustration of the basic idea, right? So in an identical twin, there's one zygote or fertilized egg that splits in two there. They can only be same sex because, again, they share the exact same underlying genomic sequence. In fraternal twins, you have, 2 fertilized eggs or zygotes, and that can create same or opposite sex fraternal twins. And so this numerical fact, right, the degree of genetic resemblance among different kinds of relatives is essential to understanding kind of the logic of behavior genetics that I hope to convey some sense of, this evening. As I was saying, identical twins share 100% of their genetic overlap. Fraternal twins or normal siblings share 50%. And as you, sort of go further out, say in a, like a family tree, the degree of overlap extends lesser, right? So for example, grandparents and so on, those are second degree relatives that share 1/4 of their genes on average. First cousins are 3rd degree relatives sharing 12.5%. Second cousins are 6.25%. And unrelated persons are predicted to share 0% of their genomic material in common because they're not biologically related to each other. So that's kind of a key sort of numerical thing to keep in mind throughout this talk. If we took random persons, say from this audience, who have no degree of biological relationship with one another and paired them, the expectation is that their psychological characteristics, their medical characteristics, their height, and so on should not necessarily be similar. And if we aggregated enough random pairs of people, right, the expectation is that the correlation among these characteristics in these randomly assorted pairs would be zero. So if we start to see systematic patterns of association or correlation in pairs of people that resemble the extent to which they share genomic material, well, that's prima facie evidence for the potential relevance of genomic factors on the similarity of people on characteristics, or what a geneticist would call phenotypes that we would be studying. So that leads to some key concepts in behavior genetics that hopefully I can convey to you that fall under the rubric of what's called ACE decomposition. So A, C, and E are numerical factors that contribute to the resemblance among people. And I'm hoping with this that I can convey some flavor of this, because I think it's an incredibly powerful set of ideas. So twin research participants provide one way to integrate nature and nurture. So as I was saying, identical twins share their DNA nucleotide sequence. You can think of them as 100% resembling each other at the level of the underlying DNA. Now, there are epigenetic effects and so on sitting on top of that, but for these purposes, we're talking about underlying, DNA variation. And again, referencing the previous slides, fraternal twins or dizygotic twins share on average 50% of their polymorphisms or different genes. So then what are these A, C, and E effects? So A refers to additive genetic effects. So this refers to the observed similarity between relatives, for example, twins, attributable to their genetic relationship, taking into account potential environmental effects. So that's key to understand, right? The logic of this kind of scientific inquiry and this kind of quantification is to take variation among people in some characteristic we want to understand. Personalities, mental disorders, height, weight, right? Anything that we're interested in trying to unravel in this fashion, and kind of separate it into these potentially different sources of resemblance among people. So again, A refers to genetic effects in these kinds of models. C refers to what are called shared or common environmental effects. So it's the distinction between A and C, right? That's part of the leverage that we get out of the twin study approach. It's the ability uniquely to distinguish between genetic effects and environmental effects. And as you'll see, environmental effects come in two varieties based on how they work, essentially. So shared or common environmental effects refer to the observed similarity between relatives that's attributable to being in the same household. So these are effects that are making people similar because they grew up together, essentially. So that's one way in which the environment might work, right? It might make people more similar because they're living in the same household, subject to the same family level influences. And that's making them similar in a specific kind of way beyond the extent to which they're similar for genetic reasons. And an example of something that's probably a shared environmental or common environmental effect might be religious traditions, for example, right? If all persons in the family are similarly subjected to these kinds of specific family traditions, then it might induce a similarity among family members beyond their genetic resemblance. But that's not the only way that the environment can operate. A third way in which, or a second way in which the environment might operate, and a third source of human variation, are unique or non-shared environmental effects. So this might be dissimilarity between relatives, net of or taking into account genetic and shared environmental effects. So these are random, perhaps even stochastic things that happen to people. So things that make you different from people with whom you might share genetic material or share environmental influences. An example might be an unpredictable accident, right? So for example, if two identical twins have roughly the same propensity for a certain psychological characteristic, but one of them is in an accident of some sort and their brain is affected accordingly, well, that's going to push the person away from whatever, their genotype is predisposing them to be like, right? For reasons that are entirely independent of their genotype, because it happened unpredictably and randomly to one person in a way that doesn't have anything to do with their genotype, right? So these are the three major effects that we can try to disambiguate by using the twin study approach. Again, genetic effects, shared environmental effects that are making people similar because they're in the same family environment, and non-shared environmental effects, those effects that are making people different in spite of the fact, right, that they might share some family environment. So now I'll say a little bit about how this works. I'm going to use something called a path diagram, and hopefully I can explain a little bit about how this works or why it's maybe a useful rubric, hopefully for understanding how this kind of science proceeds to take data, right, on psychological characteristics and turn it into inferences about genetic and environmental effects. Over here, I have a picture of André-Louis Koleski, who was a French surveyor of land, actually. But some of the mathematics underlying this were developed by him and then sort of rediscovered posthumously. So I want to sort of reference this person, right, because some of the mathematics here are linked to this thing called Koleski decomposition. So what you can see here on the left, though, is the idea of two different traits. So those could be anything that we're interested in, you know, from a human individual differences perspective. How tall you are, your stature, your weight, you know, how your weight compares to your height, essentially your BMI, right? It could be anything that we're interested in trying to understand. It could be a psychological characteristic like a personality trait. Whatever we're interested in, we can use this method to try to understand the sources of human individual differences in that trait. And in this kind of multivariate approach, we're interested not just in one trait, like the first trait, say height, we might also be interested in weight. So in this diagram, what we're saying is height and weight are two separate human characteristics that are each, you know, attributable to these three effects: genetic effects, shared environmental or common environmental effects, the C parameter, and E, the non-shared environmental effects. So what if it were the case that these two different things were associated with each other? Maybe it's the case that there's a correlation, for example, between something like height and something like weight. Maybe there's a correlation between a certain kind of personality trait and a certain kind of mental disorder outcome that we're interested in understanding. So in this augmented diagram, what we've shown is the possibility that the A, C, and E effects on the first trait might also affect the second trait. So we have the effects that are in common between the two traits, right? The A, C, and E effects on the left, and the residual effects that are unique to the second trait, the A, C, and E effects on the right. And by having those arrows that are going from A, C, and E on the left to both trait 1 and trait 2, we accommodate the possibility that some of these effects might be in common between the two traits. The possibility that we can discern something about environmental or genetic effects that drive the two traits to be associated with each other more than by chance. Now, to outline the entire model, we need to talk about the fact that there are two twins, right? So now I've added in this diagram the fact that there are two twins. Twin 1 is on the left and twin 2 is on the right. And that's because, well, twins come in pairs. So the basic logic of this kind of, you know, approach to understanding why people might differ is to study twins in pairs, right? So this might be the firstborn twin or the secondborn twin. You could also randomize twins to be twin 1 and twin 2. And the idea is that because of what we know about genetic and environmental resemblance, we can discern the influence of the A, C, and E factors, essentially, on these two traits of interest. So to do that, we have to add some of this stuff on the top of the diagram, right? So the stuff on the top of the diagram refers to how much we might expect the twins to be similar to one another under certain kinds of hypotheses about the sources of those similarity. So take a look at the A parameter and remember what we said a few slides ago about identical twins having essentially 100% genetic overlap and fraternal twins having 50% overlap. So those curved lines there refer to those coefficients of genetic resemblance, essentially. So you can see here that the A effects are correlated 0.5 or 1. So 0.5 is for the fraternal twins, reflecting the fact that they only share half their genetic material on average. And the 1 refers to identical twins who share all their genetic material on average. So if the genetic effects were the only source of variation in these traits, then those A effects should be strong. And we can estimate those A effects because we know what they should be, given what we know about genetic resemblance among twins. Now, the key thing with this is the ability to differentiate A, C, and E. So the C effects are set at 1 for twins regardless of whether they're identical or fraternal. And that's a key thing that hopefully I can convey to you, right? So that idea is that twins would resemble one another regardless of whether they're fraternal or identical, because both the identical and fraternal pairs grew up in the same set of family circumstances. So the expectation, right, if the shared environmental effect is all that matters, is that the C component should be strong, and we can estimate that expectation because it's different from the genetic expectation, right? So compare the two to each other. For the A effect, we have that difference between 0.5 and 1. For the C effect, the expectation is that it's simply 1. And it's the difference in those expectations that allows us to differentiate between genetic and environmental effects. And finally, there's the E effect, and you may notice that there is no arrow connecting the E effects. And that's because the expectation for those unique environmental effects, remember those are random things that happen to people that make them different from members of their same family? Well, the expectation if those effects matter, right, if people don't resemble each other systematically based on genes or shared environmental circumstances, is 0. And to reflect that 0, there is no correlation between these non-shared environmental or specific effects. So I realize that that's perhaps somewhat technical, but it's pretty important to understanding kind of the elegance of this research design, right? So twin studies really have this remarkable ability because of, you know, these experiments of nature that we're provided with in identical and fraternal twins to tell the difference between genetic and environmental effects. So I'll give you guys an example from some research that we did. This is in Minnesota in a twin family study. These are twins who were going into their teen years. It's a pretty big study, right? So we studied both male and female identical and fraternal twin pairs. And the thing that we were interested in here is externalizing problems. So kids acting out essentially and trying to understand, right, if things like conduct disorder, right, so that's doing some things that you might get arrested for, or being oppositional or defiant in this case, if those things were attributable to genetic or environmental forces, but also importantly, the extent to which what was going on in the parents' relationship might influence the magnitude of genetic and environmental impact. So I think this is a critical point that I want to make. The traditional approach to these kind of data would stop before this bullet point, right? It would say, so we're trying to understand why some kids are acting out, some kids are behaving more compliantly, and we're going to stop here and say, well, you know, it's such and such attributable to genetics and this percentage attributable to environments. But what we try to do in this kind of work is go a step further and ask something about the psychological characteristics of the family environment. So in this case, we knew something about whether the parents were getting along or not using a standard measure called the dyadic adjustment scale. So basically, this is asking the parents, like, how's it going at home, right? How's your marriage? You getting along well with your spouse? Or is this a source of conflict between you guys, right? Like, what is it like to be in that family? And that, I think, is a critically important sort of augmentation of this basic logic. So instead of just saying, okay, how much is genetic, how much is environmental, we're saying that those proportions might vary, right, as a function of what's actually going on in the household. Now, this next slide is for the technical aficionados in the audience, right? I'm not going to go through it in great detail, but for those of you who are, you know, interested in how this actually works, right? This is how it actually works. We have something going on in the family, a so-called moderator variable, and its impact on something we're trying to understand, externalizing behavior in this case, right? Kids acting out. And in this kind of model, it's not just the A, C, and E effects that we're estimating, but we're also going to allow those effects on this phenotype of interest, like externalizing behavior, to vary as a function of how things are going in the relationship of the parents. So there's not just these invariant A, C, and E effects on acting out in kids. Those effects are moderated, changed, shaped by the extent to which the parents' relationship is going well versus going poorly. And for those who are inclined to think about this in sort of algebraic terms, right, what we're saying is those effects, you know, like E or like A, C, and E are changed by the level of the moderator. So that's those beta terms that are in the middle. So we've augmented the model to say not just A, C, and E effects, but A, C, and E effects that might be shaped or changed by the level of the moderator. So what did we find? Well, we found evidence for exactly this kind of thing. On these graphs, what we're showing on the x-axis is the degree of parental marital discord, right? So basically, to the left is parents getting along really well with each other, and to the right on the x-axis is parents getting along really poorly with each other, on the verge of divorce, fighting all the time. On the y-axis is the degree of variation attributable again to these A, C, and E effects, right? So what we're showing here is the genetic and environmental impact on kids acting out as a function of whether parents are getting along or not. For the top two figures, we did not moderate the impact, of the parents' relationship on the kids externalizing. That's the traditional model, right? So you see there that those lines are straight because in those traditional models, right, what you're saying is genetic and environmental effects are constant regardless of whether the parents are getting along or not. Again, that's the traditional approach that is not, in our view, sufficiently nuanced to talk about the dynamics in the family and how that impacts genetic and environmental effects. That's why the lines are straight, right? It says the genetic and environmental effects are roughly, each around 30%, 30%, 30%, 30% adds up to roughly 100%, right? So they're saying those effects are very similar, regardless of whether the parents are getting along or not, okay? So now at the bottom, we introduce those moderating effects and say, well, what if we take into account whether the parents were getting along or not? Some parents are arguing all the time, fighting, high levels of parental discord. Does that change the underlying sources, right? The reasons, essentially, why kids might act out. And that's what we found evidence for in this research. So take a look at this one, for example. What we're showing here is that as parental marital discord is increasing, there's a shift between C effects and A effects, right? So on the left here, parents who are getting along really well and their kids are acting oppositionally in spite of the fact that it's a loving household, genetic effects are very important. You guys see what I'm pointing at here, right? So the A effects are more important than the C effects at low levels of parental discord. And the basic psychological interpretation, which hopefully makes some, a priori or prima facie kind of sense, right, is that kids who are acting out, even if they're in a loving family, might be genomically predisposed to acting out, right? Because they're acting out in spite of the fact that the family is well organized, loving, and so on and so forth. Now look over here, right? There's this shift at high levels of marital discord between environmental effects and genetic effects. In fact, at high levels of marital discord, these shared environmental effects are more important than genetic effects. And what that's saying is in families where kids are acting out, right? Or I should say in families where there's high levels of marital discord, the environment becomes more important. The marital discord matters, right, in terms of the kids acting out. So hopefully I've illustrated this basic point, the importance of nuance in these kinds of investigations. At the top is the traditional model. We're just going to figure out whether kids acting out is genetic or environmental, and we find that both matter. The bottom, though, adds, I think, a key degree of nuance by saying that it's not really the case that genetic and environmental effects are invariant across levels of parental marital discord. It's saying when there's high levels of discord, there's a stronger environmental effect in those families. And when there aren't high levels of discord, then the acting out is more relatively genetic. So there's this interplay, right, between genetic and environmental forces that we can hopefully discern by having this sort of more nuanced account of the data. Okay. So a little bit about now turning to the molecular world, right? So twins are one way to do this kind of work, right? And that's an amazing experiment of nature. But another way to do this is to actually look at people's genomes. So I'll say a little bit about where I think we are now in actual DNA research. So when this work started, right, this was sort of in the 90s, people were looking at what were called candidate genes. And the idea here was, well, based on various kinds of ideas about what genes might matter, we're going to pick a specific region of the genome that we think has some particular impact on the organism and try to understand it in terms of its reference to things like human behavior. And I think arguably that approach didn't work very well. It created a kind of cottage industry, right? Many studies were done like this, but ultimately those effects, that kind of research didn't pan out. So there was little success, especially in understanding sort of complex human behaviors, starting from kind of hypotheses about what genes might be relevant. So a next thing that was tried was saying, okay, those candidate genes might be relevant only in certain kinds of environments. So conceptually, it's sort of similar to the twin stuff that I was just presenting, but that didn't work out very well either, right? So the idea there was, okay, this gene is only important in these types of environments, but the genes that were studied were again picked basically in a sort of a priori fashion, right? So choosing the genes based on their putative biological relevance to a particular thing. Well, that didn't work either, right? I think that's very clear now. And again, much taxpayer money was spent on pursuing these paradigms, right, to get to this point of trying to understand how this works. And the question that I think the field faces is why, right? Why did this approach to studying, genes and environments not pan out? And I think the answers lie in both the genetic and environmental parts of the equation. So genomic variation is unlikely to be well characterized by single genomic variants. The genome, as you'll see in a moment, right, is an incredibly complex set of information, or at least extensive in any human. And these kinds of researches, this kind of approach to research, very tiny bits of the total amount of genomic variation were being studied, right? So it's like taking a little bit of information when you have all this information available and trying to understand how that little bit is relevant to something you're trying to understand. And also the environment might not be totally independent of the organism is a key environmental thing that you want to understand also. And it's that mix of things that now leads to, I think, the kind of research that's been a little more fruitful. And that goes under the rubric of genome-wide association. So as I alluded to early in this talk, right, there's been a remarkable, development in technology that, because of the Human Genome Project, essentially allows us to scan the entire genome for potentially relevant single nucleotide polymorphisms, or SNPs. I'll tell you what a SNP is in a moment, but think of it as a bit of information in a lengthy chain of information that characterizes the entire human genome. And if we're able to scan the entire genome, we're in a position to compute what are called polygenic risk scores. And the idea of a polygenic risk score is to take the information in the entire genome to indicate risk. So what I'm saying in these slides is that we've managed to move past these, single gene kinds of hypotheses to these polygene kinds of hypotheses, because we now have the technology to query or survey the entire human genome and keep the entire genome sort of in our data sets and in our conceptualizations as we're working to understand how genes might relate to things that we're interested in health and behavior and so on and so forth. Okay. So I'll give you an example of a study we did in this area. This was done under the auspices of the Social Science Genetic Association Consortium. So this is a consortium that I was involved in forming once it became clear that to do this work was going to require, fairly extraordinary resources. So when we first started doing genome-wide association kind of research, we thought we would find some things and maybe other labs would try it and find some things and we'd compare our results as science often progresses. But we quickly found that we needed bigger groups of people, bigger samples, like much more data than we initially anticipated. So that quickly led to the formation of these larger consortium efforts. So a project we pursued under those auspices was focused on well-being, depression, and neuroticism, right? So trying to understand essentially human emotional distress. And one of the things we were interested in here, so colleagues who saw me talk in psychology about my interest in classification research know that I'm interested in how those might be parts of a broader spectrum rather than separate categorical disorders. And you can see that the sample sizes here are not trivial by traditional standards of psychological research, right? So hundreds of thousands of persons in order to discern how these genetic effects might be associated with these things. And this was much of the available data in the world at the time that this study was completed. So what do we do in a genome-wide association study? Well, we're genotyping millions of single nucleotide polymorphisms across the genome. And what are those? Those are individual nucleotide bases. So DNA is this complex double helix molecule. You can see it on the side of the building over there. And the human genome consists of about 3 billion of these total bases. But many of those bases are the same between humans, right? So when we look across the genome, your DNA is the same as my DNA at the level of these individual, sort of nucleotides, right, are the codes that make up the whole DNA molecule. But every once in a while, there's a difference. Once in about maybe every 300 or so of those, there's a difference between one person and another. And just to give you some sense of the scale of this, in something that I was working on this afternoon, actually, before I finished this, talk, it's a project designed to do this kind of thing with personality traits. When we went through the whole genome and sort of figured out how many differences there were that we could then run up against personality traits, there were 16 million, nearly 17 million of these individual SNPs comparing one person to another. So you can see the challenge here. The DNA, right, of a human being is complex. There's a lot of information, right? And I mean that in sort of a bioinformatics sense, right? There's a lot of data in your genome. And what we're doing in this kind of research is very challenging because we're taking this remarkable technology, right, that just produces raw information, right, about how you might differ genomically from another person and trying to understand how it relates to something of interest. So this is a sort of basic illustration of the situation, right? There's the double helix that you guys are familiar with probably from biology class, right, that characterizes DNA. And as I was saying, every 300 or so of those base pairs, there's going to be a switch. So a SNP is person one has a particular polymorphism at that same spot as person two, right? So as you're going along the genome, people are similar, and then every 300 or so base pairs, there's a difference. So what we're doing in this research, right, is trying to take that difference, basically looking like for a needle in a haystack, right? Looking for all those differences across the genome and trying to associate them with something that we're interested in trying to understand. So this figure is something that's called a Manhattan plot. It's called a Manhattan plot because it looks kind of like a skyline. And I'll try to explain to you what it is. Across the x-axis is the physical genome laid out in sequence, right? So from chromosome 1 all the way to chromosome 22, like leaving out the sex chromosomes, so there aren't 23, but there are 22, and arranging it basically in linear sequence physically, right? So from the first polymorphism on chromosome 1 all the way down the double helix to the last polymorphism on chromosome 22, and asking if there's an association between a phenotype, right, like a psychological characteristic we're interested in, and SNPs, right? Remember those little bits of variation in the DNA in people, right? So we're taking all those little bits of SNPs and associating them with some phenotype of interest. And in this project, we did that for subjective well-being, for depressive symptoms, and for neurotic kinds of personality traits. Now, the higher the little dots float above, right, so the y-axis, larger numbers are better, right? Those are, for the statisticians in the audience, those are essentially p-values, right? So that's sort of the probability of rejecting the null hypothesis correctly, right? Or the evidence that a specific SNP is associated with a phenotype of interest. So what we're looking for in this kind of research is genomic regions that are hotspots for variation in something we're trying to understand. So if a discovery is made in this kind of research, it's because something is above like those dotted lines. And you can see that in this project, perhaps attributable to the large sample sizes, we did find some evidence for some SNPs that are associated with the phenotypes of interest, right? So there's some evidence here that certain specific genomic regions are relevant to people who might have higher neuroticism scores, more depressive symptoms, lower subjective well-being. So that's sort of important to basic biological discovery, right? So what you could do with this stuff, say, for instance, if you worked at a place like ANU, right? Like you could take this stuff over across the street to, people who study the genomes of more basic experimental animals, right? Where it's easier to characterize those regions, seek a homologue, right? Find a region, for instance, in mice, for example, that might be similar to the region in humans, and ask what it does and be able to work with it more experimentally. It's really kind of the key in a lot of ways to advances in human health and to personalized medicine, because the logic here, right, is that if we understand something about these things in humans in these very large scale studies, we can then go to the lab and kind of figure out what those genes are doing in a biological sense. Okay. But one thing that I was particularly interested in this project is something called the genetic correlation. So what we're asking here is, are the SNPs that we're finding for these traits in common, or are they distinctive? So again, for the psychological colleagues in the audience who know the work that I and others have done about classification of mental disorders, we've been very interested in the idea that mental disorders don't come in these discrete categories, but they're sort of parts of broader spectrums of human variation. So this part's particularly near and dear to me because we found that, not only is it the case that lower subjective well-being, higher depressive symptoms, and neuroticism were correlated, but that one of the potential reasons why they're correlated is because the underlying genomic signatures of those traits are similar, right? So there's not a distinction, right, in the etiology or the causes of human variation in these things at the genomic level. They're quite similar in terms of the regions of the genome that are giving rise to individual differences in these qualities. So you can see at the top, those are genetic correlations. So they're going to range from 0 to 1, 0 meaning not much of an association, 1 meaning a strong association, -1 meaning a strong negative association. Right, that there were strong associations between well-being and depression, well-being and neuroticism, and depression and neuroticism, again, suggesting genetic similarity among these supposedly distinctive human characteristics. We also ran them up against height, right? So you know how tall people are essentially. Is there a genomic correlation between how neurotic you are and how tall you are? Well, the answer is no, but that's good evidence for the genomic distinctiveness of human individual differences, right? It's not just something about the technology, right, that's indicating that these things are correlated, right? So tall people aren't necessarily more neurotic, and that makes sense genomically, according to this research, right? So the genetics of height are distinctive from the genetics of essentially being anxious or depressed and so on. Also, we looked at some other mental disorders and saw some distinction in the genomic signature between these things and those disorders, right? So there are correlations between these qualities and anxiety disorders, less so for Alzheimer's, autism, bipolar, and schizophrenia. Okay. One other, sort of empirical study before I stop torturing you with all this genomic research late on a Friday and let you go have a nice beer. But this is something that's really near and dear to my heart because I think it's really important to understand the power of genomics for clarifying the environment. So let me say more about kind of what I mean by that. I think there are signature environmental effects out there in the world, and the utility of genetics isn't just the kind of thing that I was just trying to portray to you, right? That we're going to figure out something about the genomic biology of human psychology, but also that we can kind of try to take into account genetic factors in underlying the importance of environmental effects on mental health. So this is a study that we did in the midlife in the US sample in a group of biologically unrelated persons. And what I was interested in here was the experience of discrimination, right? So if in your life you experienced a lot of discrimination in the interpersonal environment, does that impact your level of anxiety? And I wasn't just interested in, well, is there an effect? But also I was interested in, are there genetic confounds in that effect? Or is it a bona fide environmental impact, right? So what I want to know here is something like, if you've been discriminated against, does it not just make you feel more anxious, but can we show that that's actually an environmental effect, that there isn't a confound, right, of the kind of genetic risk you might have for that kind of experience? So we looked at daily discrimination, lifetime discrimination, discrimination in your job, and multiple indicators of anxiety. And we did this we controlled for genetic effects by computing polygenic risk scores. So this is something that derives directly from, the GWAS paradigm. A discovery GWAS estimates associations between the phenotypes and SNPs. There's another one of those Manhattan plots there, right? That's just an example of what we're doing. So we're figuring out how much each SNP is relevant to some trait that we're interested in, just like in the previous research I presented. And we're going to add them up across the whole genome to get an index of genomic risk for a specific person. Right, so in that kind of analysis, we know how much the SNPs matter for something of interest. And this equation just says, if we're going across the whole genome and we know how much each SNP matters, we can compute an index of risk for a particular person. So this is kind of the underlying technology for what people describe as precision medicine, right? Where the idea is, if we were successful at understanding what specific genomic variants were relevant for something of interest, you could go to your doctor, right? You could take a blood sample or even a saliva sample, use, the genotyping technology to figure out your risk for common and complex conditions that we're interested in studying. So this is stuff that's coming down the line, probably clinically useful in the next decade, I would, imagine, given how it's all going, right? Because we can predict things about your risk for particular kinds of medical conditions from your genome, essentially using this kind of approach. Now, here what I was interested in, though, was using this stuff to control for genetics, right? To show that discrimination has an impact on anxiety net of genetic risk for anxiety. So we can compute these polygenic risk scores and use them for various sorts of purposes. Okay, here's another one of these complicated looking path diagrams, but let me just run through some key things that I think we found here. On the left side is what we're using to predict the outcome, right? So on the left side is level of discrimination and social exclusion, basically the extent to which a person feels like they've been discriminated against in their life, in their daily life, over their lifetime, and in their job, right? That's what that circle is on the left. And we're using it to predict the level of negative affect and anxiety. And that shows up in things like your level of trait anxiety, how socially anxious you are, maybe even meeting criteria for a formal mental disorder like generalized anxiety, and so on and so forth. The stuff at the top is a bunch of what are known as covariates. So we're trying to take into account the stuff at the top and trying to understand the impact of discrimination and social exclusion on negative affect and anxiety. And the key thing to look at is that 0.53 in the middle, which is statistically significant because those numbers on the either side of it don't include zero. And that we've managed to estimate this taking into account polygenic risk for anxiety, depression, and neuroticism. That's those boxes at the top, along with other important kinds of things that we try to control for, like how old you are, your biological sex, your level of education, and so on. So what this is saying is there's a bona fide environmental impact, right, of discrimination and social exclusion on levels of negative affect and anxiety, basically regardless of your level of genetic risk for anxiety. So hopefully you can see the importance of this kind of thing from this kind of study, hopefully, or at least I think this is rather important, because you can use this kind of design to say the environment matters, right? There's a risk for anxiety created by social exclusion and discrimination that isn't confounded with your genetic risk for, anxiety. Because that's also a plausible mechanism, right? It could be the case that people who are anxious, tense, on edge, life is a chore, everything's unpleasant for them, feel put upon, right? They feel like they're being discriminated against. They feel like people are out to get them. And this shows up both in their subjective sense of discrimination as well as their sense of anxiety. What we're showing here is that explanation is less plausible. The more plausible explanation is that if you felt like you've been discriminated against, that has a bona fide environmental effect on your level of anxiety. Hopefully you can see the distinction between the two explanations because they're critically different in terms of how you might think about the nature of anxiety, right? Is people's sense of discrimination a function of their genetic risk for discrimination and anxiety? Or rather, is it the case that people who have been discriminated against, who've had this environmental experience, are more anxious because of that social influence? And what this suggests is there's a direct, socially mediated environmental impact of discrimination on anxiety, taking into account genetic risk for these anxiety outcomes. Okay, a few final thoughts, and then I promise you guys you can have a beer and relax on a Friday. And I appreciate your attention to all this science stuff. But a few final thoughts, right? So I think that this work involves multiple methodologies that integrate both genetic and environmental factors. So that part is very important to me, right? So I think it's really critical to not think about genes and environments as competing, right? But rather to try to articulate nuanced models that bring them together. And hopefully I've given you some sense of how we try to do that, because I really do think that that's critically important in avoiding, how these discussions tend to become polarized, right? So it's easy in this kind of thing to think of them as polar opposites, right? Either genes matter or the environment matters. But what we try to do in this work is show how they come together. And I've shown you some different approaches to that, the family and twin approaches, how to leverage contemporary molecular genetic technology to study this stuff. And I think it's very clear that those designs are complementary. So I would argue that these approaches, if they're integrated in this way, provide a lot of leverage, right? A lot of ability to better interpret the nature of human variation, and hopefully to provide a more nuanced perspective on how both nature and nurture matter when we're trying to understand very important psychological outcomes and their impact on human health. All right, thanks very much.
Bob, thank you so much for that very robust but very clear explanation of your work. And we'll just open it up to the audience for some questions and comments.
just introduce myself. I want to understand the essence of the being. I've become a neuropsychologist. And I thought I still don't understand. I became a doctor. And I thought I still don't understand. So I got into psychotherapy. And here I am. Still, I'm searching for the answer. So it's really interesting to show that modulating factors, traits, and behavior, especially with family, with behavior, with marital discord. I just want to note that, in fact, we're actually 99.9% similar, all of us, and there's only 0.1% in genetic origins and differences. So is it possible under extreme stresses that those biological processes that actually drive our primal instinct for stress probably override that small, 0.1% in our actual personality trait in the circumstances that's why we should do those effects?
Right. So let me repeat the question and also, offer my interpretation of it. I think that's a very important question. So the question is basically, given that we're very similar to each other genomically, how do we then take into account the robustness of the environment, right, and its impact on people? And I think that's the purpose, essentially, of this kind of work, right? To say that the explanation isn't solely in a kind of mechanistic biology. Right. So that's a paradigm or like a way of thinking about science that's had a lot of impact here, right? By saying, okay, even if it's a minor amount of genomic variation, that's going to provide a lot of answers for us, right? We're going to find the gene, right, that causes anxiety. That's the paradigm that I'm trying to work against, essentially, right? That it's not going to be mechanistically simple in that way, right? That if you have the gene for anxiety, then you're an anxious person. It doesn't work like that, at least according to this kind of evidence. Because the system is much more nuanced, right? And under extreme environmental circumstances, as you're suggesting, the environment can swamp out the importance of genetic individual differences. However, conversely, there are probably environmental circumstances where genes really do matter, right? So it's a matter of kind of painting that nuanced picture, right? The way in which genes and environments work together and working toward a sort of more probabilistic and less mechanistic paradigm, right? Like a way of understanding how to encompass all that information in the genome and all the complex social influences in our lives, and putting it together in a realistic way to say, these are the circumstances where genes are going to matter. These are the circumstances where the environment matters. And that leads to ways of intervening effectively, because we know kind of what the functional environments might be, net of genetic influence. So thanks for that very good question.
For example, like with health insurance companies, like using outcomes of studies for people out in healthcare based on genetic risk.
Excellent question. So the question is, this kind of research that I do and others do, in human genetics, is it possible that as we discover genomic variants that are relevant to health conditions, that information could be used in essentially pernicious ways, right? So that someone might be discriminated against for being able to obtain necessary care by virtue of their genetic risk? And the answer is yes, right? That's a risk of anything that we might do in biomedical science, right? That our research could be misused. So it's critically important that this work be done in conjunction with philosophers and ethicists and medical ethicists in particular, so that it hopefully doesn't get used that way. But I would argue, and do argue, right, that the social benefit of understanding the genomic biology has the potential to outweigh those risks if they're taken into account and properly contextualized, because many people, I think, are ultimately going to be helped by understanding more of these specific molecular mechanisms that are relevant to health. And that if we, sort of say hands off, we don't want to do that kind of research because it could be misused, we're doing a great disservice ultimately to the population by not being able to query the underlying molecular biology of conditions that are, really deadly and awful for human beings. So it's a complex question you've asked, but I think with the right kinds of, ethical oversight, the benefits to human health will ultimately outweigh the disadvantages. But we should never be complacent about what you're asking about, in my view. Other questions?
You expand a little bit on the role of epigenetics.
Yeah, an excellent question. This question was about the role of epigenetics. So In this talk, I focused solely on underlying DNA, right? So genetic differences among people. And epigenetics refers to the genomic biology that's sort of sitting physically on top of the DNA, right? So we now have technologies that allow us to characterize not just the underlying genomic DNA biology, but also things like patterns of methylation, right? So these are patterns of methyl groups that sit on top of the DNA and are associated with genes being expressed more or being expressed less. We can also directly study levels of expression, so expression transcripts, by studying RNA transcript abundance, essentially. So these are additional omics, if you will, right? Technologies that are available for querying, the genomic biology at different levels. I think that's, a critically important area that I'm personally really excited about, and tried to teach you guys a little bit about this stuff. So to add an epigenetics lecture is an additional hour, but you want to come hang out and take a class with me, we can certainly talk more about it. So I didn't have time today. I gave a lecture in this same room yesterday to colleagues in the School of Medicine about some of our epigenetic research. I think that's an area that we're very excited about. One area in particular is epigenetic age acceleration that we've been working on. So that's basically that patterns of methyl groups essentially are pretty indicative of biological aging net of chronological age. So not like how much your body has aged relative to how old you would be in terms of your time on Earth, basically. Like how old your body is in a genomic sense. And that kind of stuff is actually remarkably predictive of some interesting outcomes in circumstances where people have experienced a lot of environmental stress. So basically, right, we can sort of, using some of this epigenetic stuff, kind of figure out accelerated aging based on exposure to psychosocial stress beyond how old you say you are chronologically. Right, so kind of seeing the signature of stress on genomic biology in the epigenome. So that speaks, again, to the critical importance of environment. And that's why I'm very excited about that stuff, right? So that you're seeing in that kind of stuff that it's not just underlying DNA that's driving, important outcomes for people, but also the exposure to psychosocial stress that's driving accelerated aging. Again, speaking to why psychology matters, why the environment matters, right? That stuff is showing up in our health and well-being, again, net of underlying DNA. So I really appreciate that question. It's stuff that I'm personally very excited about in the current direction in our research program, for sure. So thank you.
I noticed that in that testimony, you said that the environment is one, but with culture travel, would be fair to assume that this is categorized differently because there will be schedules, or if they should say mortality in one thing and the other changes, and if so, how could this be measured?
Right. Okay. So let me repeat the question. The question was about basically subtleties of the way the environment might impact people and its impact on kind of the twin design. So you were referring specifically to the idea that shared environmental effects are assumed to be one, right? That if they're active, then they're making people, the same. And I think the key thing to appreciate in that regard is that it's a component of variation, right? So shared environmental effects are a kind of hypothesis about how things might work, and it's the decomposition, right, that tells you how much they're actually affecting variation. So they're definitionally one because that's the concept or hypothesis you're trying to test. So think about something like, I don't know, what they're called in Australia, zip codes in the US, like your literal location in physical space where you grew up is identical for people who grew up in the same household. So they're definitionally one. They're like shared in an obligatory sense because you can't like literally not live in your parents' house when you're young. So it's getting at those kinds of effects. What you described, right, is the possibility of like kids who are in fraternal pairs acting differently in ways that are maybe correlated with their genotype in intersection with their, sort of parental influence. That's exactly the kind of subtlety that we're trying to get at with these kinds of models. Because what we're interested in is gene environment interaction. Like in the study I presented, right, that with the parents who were fighting, right, that mattered. It's showing up in the variance components, but also gene environment correlation, right? So what if a kid who has a certain set of genetic characteristics interacts with their parents differently because they're bringing some genes to that interaction, right? It's that complexity, right, of genes and environments that we're very interested in. So I think your question is very important because that's the nuance that we're hoping to build toward, again, empirically, right? Like, how do we use data to get at that level of nuance that you're pointing towards? So thank you for that.
So I think in the interest of time, end there, but I would like to invite now Professor Paul Gerald to come and give closing remarks. Paul, the new director of the School of Medicine and Psychology. So Paul, thank you.
I have the great pleasure to close this evening and to be able to send you on your way so you can enjoy your Friday night, and to express on all of our behalf gratefulness to Professor Krueger for what I found was a really clear and lucid and very interesting talk this afternoon. This is a great pleasure to me personally. I as Bruce alluded to, relatively new to ANU. I joined here 4 or 5 months ago. I am a psychiatrist by background, so I'm intimately interested in relationships between experiences, genetics, our experience of conditions like depression and neuroticism and the like. But I'm also felt really privileged to be at this talk this afternoon because, as Bruce alluded to, this event has been going on since 2015 and actually will be the last Research School of Psychology annual lecture. It actually won't be the last annual lecture in psychology hosted by the school, but what is changing is, as Bruce alluded to, is that the Research School of Psychology is actually amalgamating with the ANU Medical School, and I have the great privilege of leading up this new entity. You might ask, why are we putting together a medical school and a school of psychology? This is very unusual. In fact, there isn't another entity like this in Australia, and I'm actually not aware of anywhere in the world where anybody has attempted to do this. But I don't think it would take much thought for you to understand what our rationale behind that is. The last couple of years has really brought to everyone's attention, you can't open a newspaper without there being some reference to it, the really indivisibility of our mental and our physical well-being and how important our mental health is to our well-being and to our society in a much broader context. And so separating those two things out, as we always have done, and having a medical school up there and a school of psychology over there somewhere, doesn't really make a lot of sense, both physically and intellectually. And so under the leadership of our Dean of Health and Medicine, Professor Gruen, and the university, they came up with what I think is a really novel, innovative, and very forward-looking initiative to bring together these two schools. And I have the great privilege, as I said, to be heading that up. And so we will continue to hold exercises like this, but it will be under a slightly different banner going forward. And Bob, you were the perfect person to I suppose, do the last of these talks under the banner of psychology, given that we're going into that, because this talk obviously was all about thinking about the interconnections and the complex and almost somewhat unsolvable interconnections between body and mind and experience, but also how we can bring modern science to try to explore some of those. And so that made your talk extraordinarily interesting and worthwhile, and I think hopefully of great interest to everybody in the audience. So thank you very, very much. We very much appreciated your time this evening, and also the rest of the contribution you've made to academic life at ANU during your visit during this week. And so thank you very, very much. And I hope everybody has a wonderful weekend.